Welcome to Season 6 of the Dementia Researcher ISTAART PIA Relay podcast. In this special series, we've invited members of ISTAART's professional interest areas to interview each other in a unique relay format. One guest becomes the next's host, and the conversation keeps moving episode by episode. ISTAART, part of the Alzheimer's Association, brings together researchers, clinicians, and professionals dedicated to understanding and treating Alzheimer's disease and other dementias. We'll be releasing one episode each day in the lead-up to this year's Alzheimer's Association International Conference, taking place in Toronto and online, showcasing the vital work of ISTAART PIAs and talking hot topics in research. Thank you for listening, and we hope you enjoy the series.
I'm Alexa Pichet-Binette. I'm an assistant professor at the University of Montreal, and I'm the education chair and incoming vice chair of the neuroimaging PIA, or Professional Interest Area. And today, I'm really delighted to be talking with Colin Groot, who is from the Atypical PIA. Hi, Colin. Very nice to see you. So as we get started, I was wondering, can you maybe introduce yourself a little bit and tell us which PIA you are involved in?
Sure thing, Alexa. I'm Colin Groot. I'm from the Amsterdam UMC, location VUMC in Amsterdam. I'm currently an assistant professor, mainly focusing on neuroimaging. I am involved in the Atypical Professional Interest Area. So it's focused around the atypical variants of AD, which I will be talking about in just a moment. I am now the current junior trainee in the executive committee, and I'm hoping to become the communications chair next year.
Fingers crossed for that. And you mentioned the work that you will be talking about more with the Atypical PIA. And can you tell us a bit more about the PIA and maybe how your research fits in that area?
I am mainly focused on neuroimaging myself. So my own work focuses on the neuroimaging aspects of atypical Alzheimer's disease. So we know from the literature that all of the atypical variants of AD are characterized by a very specific clinical phenotype. It's actually also how they are defined. But all of those clinical phenotypes also go together with a very specific neurobiological phenotype. That is to say, a atrophy pattern that's very specific to a particular variant. Now, also recently, with the addition of tau PET to our curriculum, we can also look at the tau pattern in these phenotypes, and it's also quite very different. So just to name one example, the most common atypical variant of AD is the posterior cortical atrophy. So the name already gives it a little bit away that the atrophy in this variant is specifically located towards the posterior parts of the brain. And if you look at a tau PET image of even an individual patient, so not only on a group level, but even in individual patients, you clearly see that this tau pattern is also posteriorly located. So that is my own main focus within the atypical variant sphere. It's mapping this heterogeneity in tau PET, but also structural MRI associated with each of the clinical variants. Now, in addition to that, I have a PhD student who is currently focused on, and it's actually already published in the Journal of Neurology, or I should say the journal called Neurology. Otherwise I'll get confused, which is more of a clinical perspective on atypical variants of AD. We looked at to see whether mortality from the time of a diagnosis of atypical variant of AD, so we looked at all the different variants, whether that's actually shorter than for typical Alzheimer's disease. So, and what we actually found was, yes, the time from first diagnosis to death was a little bit shorter for atypical variants of AD by from memory, I think 1 or 2 years across variants. Now, I do have to say that this is an indication that it might be that the atypical variants are progressing faster, right? So we know all this from the literature as well, that especially individuals who have a more neocortical atrophy and tau PET pattern tends to progress faster, and that's also what we see in atypical variants. However, there's also the issue that atypical variants are diagnosed typically later than typical variants of Alzheimer's disease. So it might just be that we are diagnosing people later, and that also results in a faster progression to death, let's say. But we are looking into that right now.
That's super interesting. Congrats for already having a PhD student published already also.
Yeah.
And when you mentioned this thing about the mortality, could you break down the different variants of AD, or by the different types of atypical AD, or was it more as a whole? Because of course the sample size is often quite small in those variants.
You hit it right on the nose. So that is always very difficult with the atypical variants. So the phenotypes and also the effects are quite extreme, but the samples are also always small. So just to give a quick recap of what atypical variants actually are and what we were looking at. So I mentioned the posterior cortical atrophy variant of AD, which is also called the visual variant because it starts out with visual impairments. Along with that posterior atrophy pattern that I talked about. We also have the logopenic variant of primary progressive aphasia. And the aphasia part already gives it away a little bit that it's a language issue in these individuals, at least at the beginning, mainly word retrieval and fluency issues. So that's the language variant of AD. We also looked at the behavioral variant of Alzheimer's disease in this particular study. So in those individuals, the clinical phenotype is characterized by behavioral issues such as agitation, lack of empathy, lack of motivation. So that's actually a very a clinical phenotype that's very reminiscent of a behavioral variant of frontotemporal dementia. And that's even rarer, the behavioral variant of Alzheimer's disease. So we're looking at 1 to 2% of AD cases. We were able to include a large enough sample to assess it. And then also what we looked at this, and maybe people don't know this, but there's also cortical basal syndrome that's underlied by Alzheimer's disease. So that's CBSAD in short. Which you could call the motor variant of Alzheimer's disease because it's characterized by difficulties in moving and in that sphere are the impairments. So in this particular study, we took all of them together. So the sample sizes were in the back of our head, and we looked first at atypicality, and then we clearly saw a significant difference between typical and atypical. So that's with all the phenotypes combined. But when you break them up, you clearly see that it's consistent across the different variants. But you get an issue of statistical significance because you're breaking down an already quite small group. So I would venture to say that the effect that we saw is probably across variants, although could not statistically prove it for every variant because sample sizes were just too small.
It's very nice that you could include like a broad spectrum of all the atypical variants. So that's great. I fully agree with what you mentioned about the very striking pattern of tau PET. It's a nice coincidence that we were paired together. I also look at that tau PET, and it's very, very impressive. And so you mentioned a lot of the different variants in your work, or maybe at the research center. Is there one in particular, one atypical variant that you mostly focus on, or you look at them more broadly together?
Recently, I've mostly focused on all of them together. I've mentioned this particular study on mortality. We're also looking into, and that's together with Nico Fransmeijer from Munich, into the association between network or connectivity and tau PET again. So this is also including all of the variants, and it's, I think, worth noting the results of that study as well. It's led by Hanna de Bruin, also a PhD student here. So what we saw is that taking all of the variants together, again, actually from many different centers, because we had to arrive at a sample size that we could actually do this analysis. And in Alzheimer's disease in general, there is quite a striking association between functional connectivity and tau. So functionally connected regions also tend to harbor or collect tau more strongly. So that was already done in Alzheimer's disease in general, but we wanted to see whether in atypical variants of it that would also translate, because there's quite a striking regional pattern of tau, but this is also underlined by the functional connectivity. And in short, yes, the results were very consistent across variants. So I think that's a very nice paper as well. It's under review at this moment. So that's recently. So that's mortality and the functional connectivity. But back as 8 years ago, I think I mostly focused on posterior cortical atrophy. One of the reasons is obviously because that actually is one of the most recognizable atypical variants, is also the most common. And it is not common, but it's the most common among the variants. So I think it's around 5 to 10% of cases with atypical, with AD or Alzheimer's disease. Sorry, I'm so used to that acronym, but have posterior cortical atrophy. Like 8 years ago when I did this study, there was only a sample size large enough for the posterior cortical atrophy analysis. But what we did there is actually quite different. And it's also nice to illustrate this, because there has been talk within the posterior cortical atrophy sphere that there are specific subtypes. So that there are actually within this very specific variant, there are subtypes. And the most commonly described are a dorsal variant of posterior cortical atrophy, which is to say that the tau or atrophy is mostly located towards the dorsal stream of visual processing and the ventral stream. Now, what you can expect based on those two subtypes is that the dorsal stream is more associated with visual spatial impairments, and the ventral stream is more associated with object perception deficits. So what we did is we used a method called latent Dirichlet allocation, which is a fancy name of just a specific clustering approach, to see whether, and this was on atrophy still, because it was back when tau PET wasn't very common, to see whether covariant patterns of atrophy can be detected within a sample of posterior cortical atrophy patients. And on a group level, yes, you could see that. So we could delineate actually this dorsal and ventral stream of atrophy. However, most PCA patients have atrophy to a degree in both of these streams, and there was also not a very, very clear delineation between the specific cognitive impairments that these people had. So there was I think there are some basis for subtypes of PCA, but it might be a very rare occurrence that people are really that specific into an already specific phenotype. So, but it was very nice to see all of these different factors, we called them, of atrophy within posterior cortical atrophy. But I wouldn't recommend subtyping people with posterior cortical atrophy even more, because we do know also from the literature, and this is also recognized in the published consensus criteria of posterior cortical atrophy, that it's quite a heterogeneous group. There's loads of different symptoms associated with posterior cortical atrophy, but most people have a mixed bag of these symptoms, and not a very specific set.
That's so interesting. I didn't know about these two potential subtypes within PCA, and it's very fascinating also how we can learn a lot about the brain or like disease mechanisms in a way with these atypical cases. Mm-hmm. So that's really interesting. And so now we talked a lot about the posterior cortical atrophy, and I saw that within the atypical PIA, one of the work groups is around PCA, if I'm not mistaken. Yeah. So that's also a very active area of research or of interest, I guess, within the PI. Is that right?
So there are a couple of different initiatives actually revolving around posterior cortical atrophy. One is the work group that you just mentioned, and it's basically, in a nutshell, it's the clinical criteria for posterior cortical atrophy were defined in 2017. Since then, we know a lot more, right? So on Alzheimer's disease in general, but also on posterior cortical atrophy. Mainly in terms of biomarkers that are a lot more accessible right now. So what this work group is revolving around is basically updating or providing recommendations for updating the clinical criteria for PCI in light of those new developments in the field. So that's one of the initiatives. Is led by Kier Young, who is the incumbent chair of the Atypical Alzheimer's disease PIA. And the other is a PIA-supported initiative that is led by Victoria Pellack, but focuses on the mapping the progression in posterior cortical atrophy. So there's, it's like I mentioned, that people with atypical variants tend to progress faster. But it's becoming really important to map this for the atypical variants. So that even though that's not a very common occurrence, it's still 5 to 10% of the AD or the Alzheimer's disease population. And with these new disease-modifying treatments that are becoming available, or at least trials that are now ongoing, it becomes really important to try to find a way to include these individuals into trials. And to do that, you'd need an effective method of tracking the specific progression that is seen in posterior cortical atrophy, because you have different progression of tau because it's located in other brain regions, but also the clinical progression needs to be measured differently compared to, for instance, a normal or a typical Alzheimer's disease patient, which is more language or memory focused, and in posterior cortical atrophy, like I mentioned, it's the visual impairments. Yeah. So the tracking, both on a biological level and a clinical level, need to be adjusted based on these phenotypes.
Yeah, and these are both very important initiatives. And indeed, with the incoming treatments in different parts of the world, how to establish how far along are these atypical cases will be very important.
And I should mention this also. There's a really nice. There was a meeting called CONI, Controversies in Neurology, I think, from memory, and members of the Atypical PIA were there to discuss the role of atypical AD in disease-modifying treatment trials. We had two people. One a proponent and one an opponent. Actually, the opponent was probably also agreeing with the proponent, but it was to get a discussion going. So there was a really nice event. I was not there myself, but I heard from afterwards that the general consensus was that, yes, we should definitely include people with atypical variants into disease-modifying treatment trials. And we should look, for instance, with the initiative that Victoria Pellack has started on PCA progression, look to see how we can incorporate this and accommodate their specific phenotypes.
And is this something, let's say people who are members of the Atypical PIA, anyone can join in a way if that fits their interest?
Sure, yeah.
How it is formed, or if people are thinking, oh, well, that's very related to what I do. Is it possible for people to join these working groups, or they're already kind of set?
Well, no, we are very interested in all people who want to join. And especially, I think one of the nicest things about the Atypical PIA is that, yes, we are focused on one very specific, in this case, phenotypes, but that brings together people or researchers from very different disciplines. You have a pathological perspective, you have a clinical perspective, neuroimaging perspective, genetics perspective even. We haven't talked about this, but there's also differences in genetic makeup of people with atypical variants of AD. What most obvious example is that APOE4, which is the most important risk factor for Alzheimer's disease, is less strongly involved in atypical variants. So so many different backgrounds come together in these work groups, and it's exactly what we want. Because when you're compiling these criteria, you need all of these different perspectives in order to arrive at a holistic view of a very complex phenotype. So the short answer is yes. We are very interested in for people to join. The intermission session last year, which was also last year at AAIC, which was also loosely based also on the clinical criteria, but also on we have this now. How are we moving forward? That was a very, or not very well. It was quite informal and also just nice, and to bring all these people together. So also from a personal perspective, it was nice to attend, and also professionally interesting.
This multidisciplinary aspect of the PIA is really interesting. Yeah. Mm-hmm. We tend to be sometimes more neuroimagers, but although but you're a bit more broad. That's really nice. And now you talk a lot about very hot research that you've been doing and with collaborators. But are there other, let's say, exciting areas in the field or that will be presented this year at AAIC?
So we are now selecting abstracts for the PIA session. That's every year. We have on the PIA day, which is Saturday, we have our own session. Obviously, there has been some very exciting stuff sent in. So one of the things that I do want to highlight is I mentioned the logopenic variant of primary progressive aphasia. So that's a language variant of Alzheimer's disease, and it's. Although there are quite clear symptoms that are associated with this variant, is still quite hard to diagnose sometimes, because typical Alzheimer's disease can also come with language impairments. So there are now several initiatives that are based around AI to detect speech patterns or impairments that could be indicative of a PPA syndrome in the future. So last year there was actually also some talks about that. I do have to recognize that the sample sizes for these are very small, so it's very computationally difficult to do this. The AI algorithms are still being developed, so we are not there yet. But I do think that in the future, and it's going to be a presentation on that this year as well, I know from the abstract, and also, but in the further future, I think these types of developments may become really important, because you can apply them on a larger scale. If you have a recording of someone and you can use AI to maybe get an indication or a probability that someone might get a logopenic variant PPA syndrome, this is really cool, I think. So that's one of the things that are being presented now in our incoming nice topics. And also what I found is that the co-pathology. So we know from Alzheimer's disease that many people also have co-pathology, and it becomes worse and worse when people get older. And then the co-pathology of, for instance, TDP-43 pathology, which is a different kind of pathology, is very prevalent. And the role of these co-pathologies in atypical variants of Alzheimer's disease is also an active area of research. So, for instance, this TDP-43 pathology is less commonly found in the atypical variants of AD. That might be because it's mostly related to memory impairments. So if you have memory impairments, it's usually an exclusion criteria for an atypical variant of AD. So that might be the case, but also vascular changes are less common in atypical variants of AD than compared to typical variants. So I think this is a very nice area of research, because you can use these atypical variants of AD as a disease model for what the effect is of these co-pathologies in general in AD. because atypical variants are a very nice disease model anyways, because they have so extreme phenotypes. So that is one of the things that are very interesting, and also shortens, and this is actually because it's involved in my own research here in Amsterdam. So we are, quick recap of a study that we're doing, led by Ilse Bader. We are looking at to see whether we can detect Alzheimer's disease in actually the eye clinic. So we are asking people who come to the eye clinic to do a blood test and to see whether we can detect indications of tau in the blood. But what we can also do in that same study population is do a very short test, which if you test poorly on this particular test, it's very indicative of PCA, posterior cortical atrophy. So those digitized tests that are that normal functioning people can very easily do, but people with PCA perform very poorly at. So that is very broadly applicable, I think, because it's a digital test and it's very short. So that's also a very active area of research that I'm personally interested in.
The first results of this project will be presented also this summer.
It's not developed by us, this test, so it might be that other people are working on it and are in further stages. But we are only doing the first. I think it started first a few weeks ago, even, so
nice.
Two early days.
Well, it'll be nice to follow up on that. And I've also fully agree with the hot topic of co-pathologies. Even if I work more on, let's say, more typical AD, this is also a big area of research, which pathology is more contributing to symptoms and so on. So that's a nice area of convergence. Yeah. And I was also wondering if you can, coming back a bit to the PIA, and you mentioned a lot of cool stuff that will presented in the PIA day. And could you tell us a bit more how the PIA is organized and how it works within the atypical PIA?
Like all PIAs, we have the different members with their different tasks. So we have a chair, a vice chair, a communications chair, and a programs chair, which is pretty common. Myself, I am the junior trainee at this moment. What's a little bit more uncommon is that we serve 2-year terms at the atypical PIA. So I think that leaves room for also implementation of your ideas, rather than a single year when maybe your ideas that you had one year cannot be implemented anymore because you are not elected next year. So that's nice. Also, quick shout out to the elections that are ongoing right now. So I think you can still vote for atypical AD PIAs and all the other PIAs on the ISTAART website. One of the other things that we are organizing each year, and I think we're mentioning, is we do journal clubs with the authors of the papers involved in them, which are also really nice webinars. We also organize what we call basic webinars, or actually ISTAART has named it that. But then we are taking a step back and we are looking at the really basic principles of a certain phenotype, sometimes for atypical variants of Alzheimer's disease, but also sometimes modality. So this year, for instance, we had Daniel Ohm, who was more of a fundamental scientist, focusing on the microscopic level of pathology in Alzheimer's disease and how that relates to atypicality. So I think that's always really nice, and it's also focused very much on early career researchers. And I think in that same vein, we are also very much encouraging early career researchers by only selecting early career researchers for our PIA day. We have also a poster competition where only early career researchers, such as PhD students and postdocs, can participate, or we only select the early career researchers. So I think that's one of the focuses of our PIA, is getting really those early career researchers involved in what we are doing, and we try to accommodate that by having such a broad webinars with such a broad scope. Yeah, so in that regard, we are like any other PIA where we have our FRSs that we submit each year and perspective sessions, which are also accepted for this year as well at AAIC. So,
what are the topics generally of those sessions that will be presented if people will be attending AAIC?
okay, so we have our session on the PIA day, obviously, but we have a FRS on Tuesday. Which is called, and you can put this in your AIC agenda, What is Atypical for AD? From Syndrome to Biology. And you can already garner from that title, From Syndrome to Biology, that we are shedding light on every aspect of atypical AD. We have very nice oral speakers, so Deepti Pucha, Neus Vargas Martinez, Rosalina Mohanty, who is also in our committee, and Leah Grinberg. And we are really aiming to have all of the perspective that I talked about earlier, so the clinical perspective, the neuropathological perspective, and the neuroimaging perspective all in one session. So I think if you are interested in a broad array of research, a specific focus on atypical variant of AD, you must certainly come to the session. And we have also another session that is called a perspectives session. And it's a little bit also related to what I talked about earlier, because it's called Improving Clinical Trials for Atypical Alzheimer's Disease, a call to action. So the different initiatives that I mentioned earlier, trying to incorporate atypical variants of AD into clinical trials and accommodate the different clinical phenotypes is really an area of active research, and I think it should be. So we have a great lineup of speakers there as well. It's on a Tuesday, so if you can, so if you're interested in atypical AD, you can just try to don't make it too late on Monday and come to all of the sessions on Tuesday.
Those sessions also really speak to this multidisciplinarity of the atypical PIA that we were discussing earlier. That's quite nice. And then I was thinking, is there, again, going back to this multidisciplinarity, is there, let's say, one area or one expertise that you think, oh, we would need more of that within the PIA or the field in general to like increase this dialogue within the atypical AD?
Yeah, so that's a great question, actually. So I think all perspectives are welcome, but I think the neuropathological perspective is maybe it is now spearheaded by a few people that are really great. But I think maybe it's a little bit underrepresented maybe at this moment still. So even though we are not short on expertise, we are maybe that's one area where we might have more. We might have more need for more different perspectives.
Yeah.
Yeah, and always and this is just not that there are a few people involved, but the clinical perspective is so important because it's the thing that's most important to patients, right? So that's also just incredibly important that we get different perspectives, and maybe also from countries that are maybe a little bit less represented in general. So we have people from USA and also in Canada. And also from Europe, but maybe other countries outside of those continents might be a little bit underrepresented at this moment. I think this is a feature of many of these initiatives, but we are very interested in learning the perspective of people from outside of that typical group. Yeah,
this last point really echoes a lot of also what we have been discussing in the last years within our PIA to try to increase the diversity. It's very important. And will you be presenting anything yourself or your students during the conference?
Yes, although not on atypical Alzheimer's disease. So it is relating to what we talked about, the diversity in research. So I'm this goes beyond the scope of the atypical AD topic, but I'm presenting on differences in amyloid and tau relating to different patient characteristics, such as race and age and sex. So it is relevant to atypical variants of AD, but it's not on that. And also, the PhD student that I mentioned, Hannah, is presenting on the connectivity versus tau paper. So that is very relevant. I don't remember by heart what day and time, but if you look up in the app atypical variants, you will definitely find it. So yeah, and obviously the that's not my own team, but the PIA members are presenting in the different sessions, FRS perspectives. So there's a lot going on.
Your work that you present is very neuroimaging related, so I'll mark this abstract
Exactly.
when I come to see you. Well, thank you so much, Colin, for discussing with me today. That was really great. I think now it's time to end today's podcast. But before we go, maybe a last question. As an early career researcher, would you have an advice for someone who's learning about ISTAART or wants to be involved or thought was super exciting what you presented in the PIA? So what would you recommend?
I recommend, so in the first place, if you're coming to AAIC, be at our sessions, we are always very communicative. So if you feel like you want to get involved, ask us. If you are not able to attend AAIC, we have social media channels. We are on LinkedIn, we are on X, we are on Blue Sky. We have our own page on the ISTAART website. You can even reach out to me personally if you want to become involved and you don't want to use social media. So we are very interested in all career researchers, no matter your background. So please don't hesitate to contact us via any of these channels if you want to get involved.
That's great. I think everyone should follow this advice, and everyone from the PIA is very friendly. So really, no one should hesitate, I think. Thank you so much, Colin, for taking the time to join us today. And I look forward to seeing you in Toronto.
Yeah, see you there.
Thank you, everyone, for listening. You can find profiles on myself and Colin, my very nice guest today, and more information how to become involved on ISTAART on our website at dementiaresearcher.nihr.ac.uk or also at als.org/ISTAART. And there will be a link in the show notes so you can reach those websites very easily. I'm Alexa Pichet-Binette, and you've been listening to the Relay podcast from Dementia Researcher and the Alzheimer's Association. You can hit subscribe on YouTube or any podcast app that you use to make sure you don't miss any other episode. This was the first one. We have really other nice ones coming up where you can hear more about dementia research and the upcoming conference. So thank you so much. See you next time.
The Dementia Researcher podcast was brought to you by University College London with generous funding from the UK National Institute for Health Research, Alzheimer's Research UK, Alzheimer's Society, Alzheimer's Association, and Race Against Dementia. Please subscribe, leave us a review, and register on our website for full access to all our great resources. dementiaresearcher.nihr.ac.uk.